Hello dear students, today we will discuss about the bioinformatics and its application in the pharmaceutical sector. The main objective of the current lecture is to equip the students regarding the application of bioinformatics tools in pharmaceutical sector. A plethora of genomic and proteomic information was uncovered by the US Human Genome Project, mostly by means of bioinformatics tools and techniques. Despite the fact that bioinformatics and pharmacogenomics were projected to have in the drug discovery and development process, the challenges facing the pharmaceutical industry, such as the high cost and slow pace of drug development, appear to persist. Socio-economic barriers exist that mitigate the full integration of bioinformatics and pharmacogenomics into the drug discovery and development process, hence limiting the desired and expected effects. Some of the application of bioinformatics in pharmaceutical industry are: bioinformatics is the rising sun in the global as well as for Indian pharma sector. This rising sun can produce immense economic heat for the growth of the Indian pharma sector. In the pharmaceutical sector, it can be used to reduce the time, cost, and make the people understand the complex mathematical and statistical equations in simple and easily readable form in order to facilitate drug discovery, drug development, and product development at a faster rate. Some of the important pharmaceutical areas and the utility of bioinformatics technology are: drug discovery. Only 10% of the drug molecules which are identified in research area make through the drug development phase, meaning that many potential drugs do not make it to the market and thus inflicting heavy losses in terms of time, resources, and expenses to the investors. Simulation and informatics can significantly help in improving the efficiency of drug development, cutting costs, and improving margins. There are many bioinformatic resources which are growing importance to understand and predict absorption, distribution, metabolism, excretion, and toxicity of both existing and potential drugs. Definition of a drug. In pharmacology, drug is any chemical agent that alters the biochemical or physiological process of tissues or organisms. The drug targets are usually protein molecules, and these drugs can be beneficial or harmful depending upon their effect. The main aim of pharmaceutical industry is to discover drugs with specific beneficial effects to treat diseases, especially in humans. Discovery of drug can be done by means of classical approach. This approach involves blind or hit method, also known as black box method, or classical pharmacology, forward pharmacology, or phenotype drug discovery. The origin of the use of potent compounds for the mitigation of disease conditions predates both the pharmaceutical industry and the recorded history. Historically, drugs were discovered through identifying the active ingredient from traditional remedies or by serendipitous discovery. For most of its history, drug discovery has been a product of trial and error, guided as much by the intuition and serendipity of chemists, biologists, and physicians as by any rational linear process. Thousands of chemical compounds were tested on the disease without even knowing the target on which the drug acts and the mechanism of action. 1 out of 10,000 may hit the target. Long before the pharmaceutical industry existed, drugs were discovered by accidents and their use passed down by verbal and written records. Throughout history, people have found by trial and error which berries, roots, and barks could be used for the medicinal purposes to alleviate symptoms of illness. For example, salicin from willow bark and quinine from cinchona bark. The drug discovery process, which continues to be by trial and error until early mid-20th century when the pharmacologists' basis of drugs, disease and drugs were beginning to be defined. Even then, serendipity was sought in the pharmaceutical industry as evident by the screening of known compounds or randomly testing any available molecules, for example, chlorpromazine, meprobamate, and benzodiazepines. A more rational and elongated approach became mainstream in the 1980s, that is, in vitro assay using animal tissues rather than previously used in vivo methods. This approach became central in the process for giving valuable information on structure-activity relationship and eventual pharmacophore construction. The drawbacks of the classical approach are it is tedious, time-consuming, expensive, and it is trial and error. Modern approach of drug discovery is, in modern approach, drug discovery process begins with a disease rather than a treatment. It involves the use of disease models to pinpoint relevant genetic, biological components, that is, possible drug targets. Target usually proteins, either endogenous, that is occurring within the human body, or outside agents such as viruses and other pathogens, refers to the causal agents of the disease, and lead refers to the active molecule which will interact with the causal agent. When diseases are treated with drugs, they interact with targets that contribute to the disease and try to control their contribution, thus producing positive effects. Drugs act either by stimulating or blocking the activity of the target protein. While the current practice of drug development addresses the needs of the majority of the population, scientific data has also shown that people vary in their response to the same drug. Variation between individuals in response to drugs is due to differences in their genetic makeup. Bioinformatics and the related omics technologies have been widely considered in the pharmaceutical industry, in part because they are expected to make the drug discovery process less tedious, more time efficient, more successful, and more cost effective. In fact, now there is an existence of new separate field known as computer-aided drug design. It will allow pharmaceutical firms to exploit economics of scale in experimentation through high-throughput screening. Technologies grouped under the umbrella of bioinformatics involve the use of computers to store, organize, generate, retrieve, analyze, and share genomic, biological, and chemical data for drug discovery. Bioinformatics technology is very helpful in drug target identification and validation, and in the development of biomarkers and toxicologic and pharmacogenomic tools to maximize the therapeutic benefit of drugs. The traditional chemistry and pharmacology-based approach to drug discovery has recently given way to more modernized information-based bioinformatics approach. Bioinformatics has certainly come to stay and is now ubiquitous with drug discovery. A graphical illustration of the role of bioinformatics in drug discovery is depicted in figure. One of the main thrusts of current bioinformatics method is the finding of biologically active candidates. Drugs are usually only developed when a specific drug target for those drugs action have been identified and studied. Mining the human genome sequence using bioinformatics has helped define and classify the genomic compositions of genes coding the target proteins, in addition to revealing new targets that offer potential for new drugs. By enabling the identification and analysis of more and more drugs targets, bioinformatics is expected to greatly increase the breadth of potential drugs in the pipelines of pharmaceutical companies. The current high cost of drug discovery and development is a major cause of concern among pharmaceutical companies. Along with increasing productivity, pharmaceutical companies also aim to reduce the high failure rate on the drug discovery process, so that increased number of drugs able to hit the market. The high cost of various phases of clinical trials acts as limiting factors for number of drugs which can be developed by pharmaceutical companies, and hence selecting the compounds with the best chance for approval is critical. The cost of drug discovery and development generally include total cost from discovery to approval, though some studies have included the cost of failed drugs and the costs for commercialization. Advances in bioinformatics has allowed for market efficiencies beginning with target identification and validation to assess development and high throughput screening, all with the goal of identifying new chemical entities. With more efficient target discovery and validation, bioinformatics can help ensure that more drug candidates are successful during the approval process, as well as shortening the discovery and development cycle, making it more cost-effective. Steps involved in drug designing. The processes of designing a new drug using bioinformatics tools have opened a new area of research. In order to design a new drug, one needs to follow drug target identification. What protein can we attack to stop the disease from progressing? Drug development begins with the identification of a suitable target, which must contribute significantly to a human disease. Drug target validation. A target needs to be evaluated to ensure that modulation of the target will have desired therapeutic effect. This process can be accelerated with bioinformatic tools, therefore speeds up the target validation steps significantly. Lead identification. High throughput screening is a major strategy for the discovery of new leads. With bioinformatics, protein targets are screened against database of small molecules compounds to see which molecule binds strongly to the target. A unique lead molecule should possess following desirable features, that is shown in figure. Lead identification can be done by means of serendipity, by prospection of natural resources, large-scale screening of related compounds, high-throughput screening. Lead optimization. Those molecules fulfilling the lead identification criteria can go to molecular finishing school. At this stage, the computational technique used in refining the structure of lead compound is QSAR, that is, quantity structure activity relationship, to improve the potency and selectivity. The information from QSAR can be used to suggest new chemical modifications for synthesis and testing. Preclinical and clinical development. Once a drug has been shown to be effective by an initial assay technique, much more testing must be done before it can be given to human patients. Animal testing is the primary type of testing at this stage. Eventually, the compounds which are deemed suitable at this stage are sent on to the clinical trials. In the clinical trials, additional side effects may be found and human dosage are determined. Clinical trials are designed to determine safety and tolerance levels in humans and to discover how the drug is metabolized. Search programs. Before starting laboratory-based screening experiments, it is always better to generate as much information as possible about potential drug-target interaction. Bioinformatics tools play a pivotal role in screening the target molecules and their interaction with a particular drug. Algorithms can be used to identify potential interacting ligands based on the goodness of fit structure of a target protein is known, thus allowing rational drug design. Nowadays, various docking algorithms have been developed which attempt to fit small molecules into the binding sites using information constants and bond energies. Molecular docking. It is a process by which two molecules are put together in 3 dimensions to find binding modes of protein with ligands inhibitors. In molecular docking, we attempt to predict the structure of the intermolecular complex formed between 2 or more molecules. Docking algorithms are able to generate a large number of possible structures. We use force field-based strategy to carry out docking. The docking process involves 2 basic steps: prediction of the ligand conformation as well as its position and orientation within these sites and assessment of the binding affinity. These 2 steps are related to sampling methods and scoring schemes respectively. Various docking algorithms used in drug designing are dock and combi-dock. Algorithms are available that helps compare 2D or 3D structure analysis, that is X-ray crystallography, NMR, and build a profile of similar molecules. Next is active site analysis. Studying the active site of a target molecule is a prerequisite to develop a lead compound via computer softwares. The surface of the target molecule to be interacted by lead may have various chemical environments such as hydrophobicity, hydrogen bonding, or catalytic zone. To this field, fragments of a hypothetical compound is placed. The orientation of the fragments provide a clue about the final form of the lead compound. Currently, various softwares are employed for this purpose such as GRAIN, History, Hint, Grid, and Buckets. Initially, a prototype molecule is positioned inside the active site to satisfy a few of the bonding energy. Additional building blocks are fitted to stepwise manner till all the bonding energies are satisfied. LIX is a software that creates the active site points and then searches for chemical structure database that would satisfy the active site. Popular drug target database. The various popular drug target databases are Potential Drug Target Database, Drug Bank, Therapeutic Target Database, TDR Targets Database, Matador, TB Drug Target Database, Drug Board, CHAMBL. Promote novel new drug development. There are some collateral costs that bother the pharmaceutical industry. These costs include commercialization cost, litigation, and drug recall cost and general cost to society. Commercialization costs of new drug to be about $250 million per approved drugs are high, mainly because most new drugs approved are essential functional replicas of the drugs that already exists. Most of the copycat drugs are being commercialized to handle the illness for which there are drugs already. Thus, there is a need of interface that can attract the attention of both physicians and patients who already have access to similar medication. Bioinformatics can act as proper interface and provides new approaches and opportunities to pharmaceutical companies to efficiently discover potential drug targets and develop novel drugs. If drugs are not commercialized in competition with already existing equivalents, their commercialization costs are expected to fall significantly. Product formulation designing. Formulation is a process of mixing of ingredients in such a way as to produce a new or improved product. The formulation department must balance the different marketing and deliverability requirements with chemical constants and cost to come up with the best possible drug delivery method at the best price. The information regarding the number of batches which are formulated in the designing process will be stored in the corporate database, which will help the scientists to assess the results and methods from anywhere in the world. They can use their time and resources on new research rather than repeating the old work. Also, with the aid of optimization softwares, the best formulation can be obtained by using the data of the previously formulated batches. Pharmacology. Bioinformatics is used in drug target identification and validation, and in the development of biomarkers and toxicogenomic and pharmacogenomic tools to maximize the therapeutic benefits of drugs. Examples where bioinformatics has been utilized: discovering novel carriers for oral insulin tablets, a pharmacoinformatics approach. Insulin is used medically to treat type 1 diabetes mellitus, most commonly injected subcutaneously for human beings. The realization that insulin injections have become a part of life can be extremely harrowing for many diabetic patients. Oral delivery of insulin in tablet form has always been a significant challenge for pharmaceutical researchers. This study is a prospect of oral insulin tablet through pharmacoinformatics approach. Molecular medicine. The human genome will have profound effects on the fields of biomedical research and clinical medicine. Every disease has a genetic component. This may be inherited, for example, cystic fibrosis, Huntington's disease, or as a result of body's response to environmental stress, which cause alterations in the genome, for example, cancer, heart diseases, and diabetes. The completion of the human genome means that we can search for the genes directly associated with different diseases and begin to understand the molecular basis of these diseases more clearly. This new knowledge of the molecular mechanism of disease will enable better treatments, cures, and even preventative tests to be developed. Personalized medicine. Personalized medicine is the use of diagnostic and screening methods to better manage the individual patient's disease or predisposition towards disease. Personalized medicine will enable risk assessment, diagnosis, prevention, and therapy specifically tailored to the unique characteristic of the individual, thus enhancing the quality of life and public health. Dear viewers, with this we conclude this very important topic of today. Hope you enjoyed the lesson. Goodbye. Stay blessed.